Hello and welcome. My name is Julie Eakins. I am a Senior Research Scientist here at Cyprotex. I would like to introduce you to some of the early in vitro mitochondrial screens used in drug safety. The aim of this presentation is to describe a number of in vitro approaches to identify compounds which interfere with mitochondrial function and why we believe this is important. In order to achieve this, I will give a brief introduction to the area of mitochondrial biology and toxicity. I will then describe a number of common in vitro assays. These will include both the simpler cytotoxicity assays, along with the more complex assays using the adjuvant seahorse flux analyzer. Using these assays, I will demonstrate how it is possible to not only enhance the prediction of mitochondrial toxicity, but also gain an insight into possible mechanisms. Following this, I will look at mitochondrial toxicity and its role in drug-induced liver injury, or DILI, one of the most frequent sources of compound attrition in both early and late-stage drug development. Finally, I would like to present a short case study using the recently withdrawn from clinical trials TAC 875. As you may be aware, mitochondria are responsible for a variety of cellular functions, their major function being the production of over 95% of cellular energy in the form of ATP, via a process known as oxidative phosphorylation, or OXPHOS. In this process, energy fuels such as glucose, fatty acids and proteins are metabolized in the presence of oxygen to ultimately form water, carbon dioxide and ATP. This uses a series of enzyme complexes and pumps known as the electron transport chain, or the ETC. However, mitochondria are also involved in a number of other critical cellular processes, including apoptosis, calcium flux and homeostasis, cell signaling, fatty acid metabolism, heme synthesis, and cholesterol and hormone synthesis. It is clear that these organelles are essential for normal cellular function. The importance of mitochondrial health is evidenced by the fact that numerous diseases and metabolic disorders are associated with mutations in either mitochondrial DNA or nuclear DNA which encode for mitochondrial proteins. Examples of these metabolic disorders are Leigh syndrome and Leber's hereditary optic neuropathy. There is also growing evidence that mitochondrial dysfunction is associated with both dementia and neurodegenerative disorders. This has been demonstrated by Cannon et al., who described the development of a preclinical model of Parkinson's using the well-documented complex 1 inhibitor rotenone, all of which demonstrate that disturbing mitochondrial function can have deleterious effects, which leads nicely into the main topic of today's presentation, that of mitochondrial toxicity. Mitochondrial toxicity has been considered an important mechanism of toxicity for a number of years. It's been implicated in high-profile withdrawals from the market, such as triglycerone, tolcapone and cerevastatin, along with a number of black box warnings. It has been implicated in multiple organ toxicities, including liver, cardiac, kidney, as well as a potential contributor to idiosyncratic DILI. All of this implies that the early detection of compounds which affect mitochondrial function would be beneficial in drug discovery. As such, there are a number of approaches which have been developed, which can be incorporated into early drug discovery screens. Here I will present some of the typical in vitro approaches that have been established, which I will take you through in more detail. Firstly, the glue-gal assay. This was one of the earliest screens described and was first published in 2007 by Mariquin et al. In this assay, the cytotoxicity of the compound is determined under two different culture conditions, glucose and galactose. Under standard laboratory conditions, it is common to culture cells in moderate to high glucose media. When using transformed cells such as EpG2s, this encourages the cells to rely on glycolysis independent of the mitochondria. This is known as the Crabtree effect. By exchanging the media to one containing galactose, this forces the cells to rely on mitochondrial OXPHOS to supply ATP, rendering them more susceptible to mitochondrial toxicants. In the glugal assay, the test compound is dosed in media containing either I-glucose or galactose. Following a 24-hour incubation, cytotoxicity is assessed using cytotoxicity endpoints such as cellular ATP or the MTT assay. Using a reference set of compounds, we established that if the compound is greater than threefold more toxic under the galactose conditions, this implies the compound is a mitochondrial toxicant. An example of which can be seen here, where the cytotoxicity of entacapone is 10 times more potent in galactose media compared to glucose media. We can also assess mitochondrial health using eye content imaging to determine the effects of compound on the mitochondrial membrane potential. In these assays, you can use a number of mitochondrial dyes, such as rhodamine or TMRE. These dyes are both potentiometric dyes, which are sequestered into the mitochondria when there is an active mitochondrial membrane potential. In active mitochondria, the dye is taken up, and you can measure the amount of dye taken up into each cell. If there is a lower MMP, then the dye is no longer sequestered, and we get a lower MMP signal. In the example shown, HG2 cells were dosed with either vehicle control or 5 nanomolar rhodamine for 24 hours, and using eye content imaging determined the cell count by using Hurst and mitochondrial membrane potential using rhodamine 123. In the upper panel, you can see that the cells are stained for both nuclei, blue colour, and associated with these is mitochondrial rhodamine staining in green. With 5 nanomolar rotenone in the lower panel, there is no cell loss. However, the associated rhodamine signal is much reduced, indicating a loss in MMP. Also, you can see in the table some reference compounds. Using this high content imaging approach, we established that a twofold shift between MMP and cell count loss resulted in the successful identification of the majority of compounds. Now a brief introduction to the mitochondrial stress test assay. As mentioned previously, this uses the XFE96 flux analyzer, which uses cartridges with solid-state sensors to simultaneously measure oxygen and pH. This allows the measurement of oxygen consumption rate, OCAR, and extracellular acidification rate, ECAR, in real time, a determinant of OXPHOS and glycolysis, respectively. Before I proceed, I will quickly explain the terminology in more detail. Oxygen consumption rate, OCAR, is a measurement of extracellular oxygen content. As mentioned previously, oxygen is required during the OXPHOS process. Thus, a decrease in the rate indicates reduced mitochondrial respiration. Although increases in OCAR can also be seen with a class of compounds known as uncouplers. Extracellular acidification rate, ECAR, is a measurement of extracellular pH. The dominant source of hydrogen ions is lactate. Hence, an increase in ECAR is deemed as an increase in glycolysis. This is seen when mitochondrial respiration is inhibited and is an adaptive response to the reduced ATP formation via OXPHOS. The final term is the reserve capacity. This is the ability of the cell to respond to an increase in energy demand. A reduction in this feature indicates mitochondrial dysfunction and demonstrates how close to the bioenergetic limit the cells are. In order to generate these measurements, we use the 4-injection port system on the seahorse cartridges to sequentially add our test compounds and selected inhibitors to the cells to determine basal OCAR, ECAR, the maximum respiration rate and reserve capacity, ATP-linked respiration, proton leak, and non-mitochondrial respiration. Initially, 4 measurements of OCAR and ECAR are taken to determine the baseline. Then the test compound is injected directly onto the cells, and OCAR and ECAR measurements determine the basal effects. The stress tests are then performed by sequentially injecting oligomycin, FCCP, and finally rotenone and antimycin A. These are known inhibitors of the ETC. Injection 2 delivers the oligomycin, a known inhibitor of ATP synthase, also known as complex 5 of the ETC, which is responsible for the phosphorylation of ADP. By inhibiting complex 5, this prevents the ETC from functioning, and subsequently a decrease in OCAR is observed. This allows the determination of ATP-coupled respiration. Injection 3 delivers FCCP, which is a mitochondrial membrane uncoupling agent, allowing the free movement of the ETC. However, this does not generate ATP, since complex 5 has been inhibited, and consequently this results in the maximum ETC activity and allows the detection of the cellular maximal OCAR and the spare reserve respiratory capacity, which is termed as the reserve capacity. Injection 4 delivers the addition of rotenone and antimycin A, abolishing the ETC by blocking both complex 1 and 3. This inhibits all mitochondrial OCAR and allows the degree of non-mitochondrial respiration due to non-mitochondrial oxygen requiring enzymes, and allows the calculation of the proton leak. Here we show some example data with some typical effects on OCAR, reserve capacity and ECAR. In panel A, you can see an example of a compound which increases OCAR. This is seen in response to 2,4-dinitrophenol, a known mitochondrial uncoupler. Panel B shows the effect of a compound which results in a dose-dependent decrease in reserve capacity, but interestingly has little effect on basal OCAR, such as seen with diclofenac. Panel C shows a compound which showed a dose-dependent increase in ECAR, in this case triglytazone. And finally, in panel D, we show a compound which decreased both OCAR and reserve capacity. This example is fenformin. As the mitochondrial stress test generates bioenergetic profiles, i.e. changes in OCAR, reserve capacity and ECAR, this allows the identification of potential mechanisms. Here we show some pictorial representations of how we would normally expect these bioenergetic profiles to look like for some mitochondrial toxicants. In the upper left diagram is what we would expect an inhibitor of the electron transport chain, such as antimycin A, to appear as. With inhibition of the ETC will decrease both OCAR and reserve capacity, with an increase in ECAR to compensate. Whilst compounds which uncouple oxidation and phosphorylation, the lower left diagram, such as 2,4-dinitrophenol, an increase in OCAR and a compensatory increase in ECAR would be expected, with no effect on reserve capacity as the cells will already be at a maximal respiration. For ATP synthase inhibitors, upper right diagram, such as oligomycin, we would expect, like the ETC inhibitors, a decrease in OCAR with the concomitant increase in ECAR. However, we would not expect any effects on reserve capacity, as this feature is independent of ATP synthase activity. With general cytotoxic compounds such as verapamil, we would expect a decrease in all three features: OCAR, reserve capacity and ECAR. Using these blueprints, we were able to assign unknown compounds with potential mechanisms of action. In 2016, we published our findings, in which we used a reference set of over 70 compounds. All compounds were tested to the limit of solubility and covering a range of mechanisms, although, as you can see, they were predominantly ETC inhibitors and uncouplers. We analysed the bioenergetic profiles of the complete compound set using an unbiased approach. Whilst not definitive, you can see there is some concordance between the actual and predicted mechanisms. However, as you can also see here, that we had also identified a number of false positive compounds. As the compounds had been tested to the maximum solubility, we looked at the effect of normalising the data to see if we could improve the sensitivity and specificity of the assay, as covered in the next slide. Therefore, For both the glue-gal and the seahorse assay, we looked at the effect of normalising the data to human plasma concentration, Cmax. In order to achieve this, we took 59 drugs and chemicals, all with known human Cmax exposure data. These included 27 known positive compounds, covering different mechanisms, and 32 negatives, either with toxicity associated, but with no known mitochondrial component, or with no known toxicity liability. When we normalise the data to 100 times Cmax for seahorse, or as a combination with a threefold shift in cytotoxicity for the glue-gal assay, as well as with the 100 times Cmax, both of these assays showed no false positive compounds, and as such gave 100% specificity. The seahorse assay correctly identified 21 of the known mitochondrial toxicants, giving a sensitivity of 78%. The glue-gal demonstrated lower sensitivity at 41%. By combining the outcome from either assay, the specificity remained at 100% as expected. However, the sensitivity increased to over 81%, and the accuracy of this approach increased to over 91%. This was due to the fact that the glue-gal assay picked up one compound not determined in the stress test. Following on from the seahorse assay, we have the permeabilised cell ETC complex assay, again using the XFE96 flux analyzer. The aim of this is to gain a further understanding of the mechanism of toxicity for ETC inhibitors. The assay is performed in a single well and involves, as suggested, the permeabilisation of the cells, in this case EpG2 cells, and looks at the recovery of OCAR inhibition. The cells are initially assessed in the presence of pyruvate and malate substrates for complex 1. And mitochondrial respiration, OCAR, is determined. If in the presence of the compound OCAR is inhibited, this indicates an inhibitor of either complex 1, 3 or 4. No inhibition of OCAR still remains the potential to be a complex 2 inhibitor. Rhodamine is then added to block complex 1 and any reverse electron transport, and along with the succinate, the substrate for complex 2. If OCAR recovers to vehicle, this shows that the complex 1 was inhibited. If no recovery is seen, this suggests this is either complex 3 or 4. If inhibition is seen, which was not present before the addition of succinate, this identifies complex 2 inhibitors. Finally, if in the presence of ascorbate and TMPD, with antimycin A to block complex 3, if recovery is seen, then this indicates a complex 3 inhibitor, with continued OCAR inhibition indicating complex 4. Here are some examples of the data generated using compounds with clearly defined mechanisms of inhibition. In the panel A, the complex 1 inhibitor rotenone inhibits pyruvate respiration, but as expected, recovery of OCAR was seen with the complex 2 substrate succinate. Panel B shows the effect of thiaflutamide, an inhibitor of complex 2, in which we only see inhibition of succinate respiration. This is an important aspect as it allows differentiation of the complex signals. Recovery is observed with ascorbate addition. Panel C shows the response to antimycin A, the complex 3 inhibitor. In this example, we see inhibition in the presence of both pyruvate and succinate substrates and show recovery with ascorbate. Panel D, sodium azide, a complex 4 inhibitor, in which we see inhibition of all 3 substrates, i.e. no recovery is observed. Finally, we have the negative control betaine, in which no inhibition was seen. Therefore, the sequential inhibition recovery allows us to identify which complex has been inhibited. Using a set of reference compounds with literature-defined modes of action reported to inhibit single complexes of the ETC, we assess these in both the stress test and the permeabilised cell assay. Using the stress test, all of the compounds were identified as having some mitochondrial effects. What may be apparent from this data is that whilst the majority of the complex 1, 3 and 4 inhibitors were correctly classified as ETC inhibitors, only one of the complex 2 inhibitors was assigned the correct mechanism. Following indications using the stress test with the permeabilised cell assay allows us to further interrogate the mechanism, as shown here. The majority of the compounds were then identified with the corresponding predicted complex inhibition. Although the complex 2 inhibitors were identified as mitochondrial toxicants in the stress test, the mechanisms are difficult to detect in intact cells, possibly due to complex 1 preference in high substrate environments. However, using the permeabilised cell assay, the majority of the compounds were correctly identified. And by combining the stress test and the permeabilised cell assay, we can distinguish between the complex 4 and complex 5 inhibitors. We have recently assessed a further reference set of compounds consisting of 52 drugs which we tested up to 100 times total plasma Cmax. In this dataset we determined the glue-gal shift, the mitochondrial stress test and the mitochondrial membrane potential as described earlier. This set consisted mainly of compounds with known organ toxicities, 23 of which were associated with mitochondrial toxicity and 29 not associated with mitochondrial toxicity, of which only 4 of these had no reported organ toxicity. As you can see here, once again, neither the glue-gal nor the stress test identified any false positive compounds, confirming the findings of our earlier publication. Although this is a different reference set of compounds, once again the stress test gave the highest sensitivity, 87%, also as previous. The MMP assay appears to be the least specific with 7 false positives. However, this data set included a number of cytotoxic compounds, and it is possible that we may be detecting cells undergoing apoptosis or indirect mitochondrial responses. We then followed up on these mitotoxicant compounds using the permeabilised cell assay to assess the potential mechanism. In this table we have the suggested mechanism according to the literature, the predicted mechanism according to the stress test, followed by the permeabilised cell assay mechanism. The majority of the compounds were identified by one or more of the in vitro assays, and there was good alignment with the literature described mechanism. Some of these compounds do have mixed mechanisms which are difficult to determine using a single assay, but are aided by the combination of the stress test and the permeabilised cell assay. There is a lot of data here, but if I can draw your attention to one example, flutaminide, in the middle of the table. This is an hepatotoxic compound associated with inhibition of complex 1. This was positive in all 3 of the in vitro assays and was predicted via the stress test to be an ETC inhibitor. Using the permeabilised cell assay we demonstrated that indeed this was a complex 1 inhibitor. Interestingly, paroxetine and promethazine were not detected in any in vitro assays, which could indicate either these are not mitochondrial toxicants or are cell-specific responses. As mentioned earlier, mitochondrial toxicity has been implicated in a number of drug withdrawals. Drug-induced liver injury, or DILI, is one of the most common causes of preclinical, clinical and post-marketing attrition. Based on this, we assessed a number of known hepatotoxic DILI compounds and non-DILI compounds, again using the glue-gal assay, mitochondrial stress test and the MMP assay. The boxes highlighted in red are compounds which resulted in a positive signal in any of the assays. The boxes in yellow show the sensitivity and specificity for each assay. Once again, the glue-gal and the stress test identified no false positive compounds. The stress test alone identified that 39% of the DILI compounds had a mitochondrial liability, and when we combined both the glue-gal and the seahorse assays, as we had seen previously, the sensitivity increased to 42%, since one compound, ritonavir, was identified in the glue-gal but not seahorse. This data suggests that mitochondrial toxicity is involved in over 40% of DILI compounds. This is supported in a recent Pfizer paper in which Rana et al. Earlier this year published similar findings. They reported 45% of DILI compounds were associated with mitochondrial liabilities. Where this dataset and that of the Rana publication matched, we saw that 19 of the DILI compounds and 8 of the non-DILI compounds had the same mitochondrial call. Finally, I would like to introduce you to a case study using the compound TAC875 of Fazigligam. TAC875 is an oral selective GPR40 agonist that was in development by Takeda for the treatment of diabetes. It was voluntarily withdrawn in phase 3 clinical trials due to liver safety signals, DILI. There was no evidence of rat in vivo toxicity, although some evidence was seen in dogs at high doses. The in vitro data, such as the glugal assay, had shown no evidence of mitochondrial toxicity, although inhibition of the hepatobiliary transporters BSEP and MRP2 had been observed. We collaborated with Utilising to investigate the mitochondrial liability further. Utilising are a simulation company, with Cyprotex being the preferred provider for high-content imaging and mitochondrial assessment data. Early data published by Francis Walensky et al. In 2017 had suggested TAC875 was not a mitochondrial toxicant due to no response in the glugal assay. This was initially confirmed when we saw that TAC875 did not appear to shift cytotoxicity, irrespective of the endpoint chosen. These assays were performed in the presence of serum, as in the early publication. However, TAC875 is highly protein bound at 99.8%, with a total plasma Cmax of 10 micromolar, giving a free plasma concentration of 14 nanomolar. As such, the glugal assay was performed in serum-free conditions, using again a variety of cytotoxicity endpoints. As can be seen, there is now a clear shift in cytotoxicity in galactose media compared to glucose, with a fold shift 3 or more, irrespective of the end point. We then assess the effect of TAC875 on the mitochondrial membrane potential over a time course, either in the absence or presence of serum. The upper graphs show cell count on the left and MMP on the right under serum-containing conditions. The lower graphs are the serum-free conditions. As you can see, even in serum-containing media, there is a decrease in membrane potential following 6-hour incubation. Under serum-free assay conditions, the decrease in MMP is seen after only 1 hour incubation. There is a maximum 21.7-fold decrease in MMP signal compared to cell count at 6 hours, and this ratio drops over the remaining incubation period as cytotoxicity is evident. TAC875, in summary, causes an inhibition of the mitochondrial membrane potential and is seen over multiple time points and under both assay conditions. The response is earlier and stronger in the serum-free conditions. We also assessed the effect of TAC875 on mitochondrial function using the stress test again over time. We either added the compound directly to the cells as already described, so that's 0 hour, or we pre-incubated for 1 or 24 hours prior to measurement of OCAR, reserve capacity and ECAR. Inhibition of OCAR and reserve capacity was seen at all time points, and the AC50 was below 100 times total plasma Cmax at all time points. Following a 24-hour incubation, both OCAR and reserve capacity showed significant decreases with minimum effective concentration values, the point at which the compound response falls outside of the vehicle control, was of 0.19 and less than 0.1 micromolar respectively. The earlier time points indicate an ETC inhibitor leading to cytotoxicity at the later time point at the higher concentrations. One point of note is that the mitochondrial stress test does not contain serum. Following on from the stress test, we performed the permeabilised cell assay to confirm the ETC inhibition. The data generated suggests that TAC875 is a complex 1 inhibitor. The compound was tested to the limit of solubility. Although the response observed in the permeabilised cell assay is not as strong as that seen in the mitochondrial stress test, which is likely due to the BSA which is present in the assay to help maintain fully functional mitochondria. We ran a comprehensive set of assays which is detailed in the table, but have not all been covered in this presentation. In summary, the seahorse accurately identified TAC875 as a mitochondrial toxicant, predominantly exhibiting electron transport chain inhibition, with values for both OCAR and reserve capacity being less than 1 micromolar at both 1 and 24 hours. Measurement of mitochondrial respiration using the stress test was the most sensitive assay in EPG2 cells. The stress test findings were also confirmed in both primary human and rat hepatocyte, again with values being within the total plasma Cmax value of 10 micromolar. Protein binding may explain the lack of observed mitochondrial toxicity in the glugal assay in the presence of serum. On removing serum, a glugal shift was observed. We recently published a joint publication with Utilising and Takeda using some of this data. Finally, I would like to summarise the findings I have presented here today. Early discovery screening for mitochondrial toxicants would be beneficial and can be performed using a number of different in vitro approaches. Both the MMP and the glugal assays are amenable to 384 well format, unlike the XF flux analyzer based assays, and both have shown high specificity, in particular the glugal assay. The stress test does show the highest sensitivity towards mitochondrial toxicants. Using a combination of both the stress test and the permeabilised cell assays can provide insights into the possible mode of action. The stress test can indicate if a compound is a mitochondrial toxicant and can also indicate potential mechanism. This can then be confirmed by the permeabilised cell assay, which can be used to determine ETC inhibition and associated complex inhibition. This mechanism of action information could be useful, especially during early drug discovery, aiding chemical selection. We have shown that DILI prediction using mitochondrial toxicity as a single mechanism with 100% specificity and 42% sensitivity when combining the glugal and stress test, illustrating the importance of mitochondrial liabilities in DILI. Using a multiparametric approach, I identified TAC875 as a mitochondrial toxicant and as a complex 1 inhibitor. Hopefully we have also demonstrated that this in vitro toolbox approach is valuable in predicting mitochondrial toxicity potential and mitigating this risk in organ toxicity. I would like to thank you for your attention. If you would like to find out more about these assays or any other assays, we can be contacted via our website www.cyprotex.com, or you can contact us at enquiries@cyprotex.com. Thank you.